Hello, my name is Brian O'Connor. I'm the software architect and owner of Nimbus Informatics. Nimbus is a consulting company that specializes in helping researchers analyze their next-gen sequence data on Amazon's cloud. This is the third part of a three-part series of video tutorials on how to analyze data through our Nimbus portal web application. In the previous two videos, I showed you how to set up your studies, samples, and experiments in the portal, and then I showed you how to upload data and associate it back with those samples through the web application and using our upload tool for transferring data into the cloud. In this tutorial, we're going to take those two FASTQ files, which are each one of a pair of FASTQ, a paired end read, and take those and use those as the input for our Nova-Align workflow and then subsequent workflows as well. Okay, so I'm going to go to portal.nimbusinformatics.com. If you're not already a customer and not set up, please email us at help@nimbusinformatics.com and we can help you get set up with an account and talk to you about your needs for your computational analysis needs on EC2. Okay, so I'm going to go ahead and just log on here and click Login. And I'm logging in as an admin user, but you can log in as your own user. It's virtually the same. Now, the last time we set up this test study, and I associated an example experiment and a series of files with this sample. So in this case, you can see it's a paired end read. These are the two files that are independently represent the reads in the paired end read. These are gzip FASTQ. This is the preferred format we like for our workflows. It'll minimize the amount of data transferred up to the cloud, and these are Sanger scaled. Okay, so what I'm going to do is I'm going to move over out of the Studies tab and go into the Analysis tab over here. I'm going to go ahead and click on Launch Workflows, and the first workflow I want to launch is going to be a variant annotation workflow, just to show you what the end of the process looks like. So I'm going to click here, I'm going to click on this, and what's happening now is it's searching the database and it's finding all of our VCF files that we've produced. This is the variant calling file. So I can drill down into some previous experiments that I've done, and what you'll see is that as I drill down into it, you can get a sense of how our overall workflow works. So you can see here this is a previous upload, and underneath the upload we have the gzip FASTQ quality report and filter, which is basically just filtering and generating a quality report for us. So if I scroll down a little bit, you can see that I've then taken this file and applied Nova-Align to it. And then if I drill down into Nova-Align, you can see the individual steps of the Nova alignment workflow. So the nice thing about these tree structures is it really gives you a very clear idea of the individual steps of our workflow and how everything relates to each other. Since it is flattened out into this tree structure, there are duplicated nodes in these trees, but that's good because it actually shows you the rich sort of parent-child relationships where you might take multiple samples and they're all the parents of the same analysis output. So that's normal to have duplication in this tree structure. But you can see here now I've run a workflow for GATK recalibration and variant calling. This one's particularly tuned for exomes, and inside that there's a bunch of different steps that'll eventually take me down into the actual variant calling. Now in the future we're working on right now an abbreviated version, so in addition to this tree view we're working on a collapsed view that just gives you a high-level overview of the output files for each workflow, and that'll actually reduce down the amount of clicking that's necessary. You'll probably switch back and forth between the two views because frankly having the tree structure is actually very, very helpful for seeing how the workflow works. Okay, so in this case what we see here is a merged VCF file. This contains all of my variant calls for this particular series of analytical steps. We've done alignment, variant calling, and this is the output for that. So I'm just going to go ahead and select that. Now what happens is if we scroll all the way to the top, and I'm just going to collapse this, you can see that this is now the selected input for my variant annotation workflow, and I can click Next. Okay, at this point this is a summary page that just tells you this is what we're going to do. It's a description of the workflow. Now this workflow is a variant annotation workflow, and this is applying OMEM, GWAS overlap, EvoFold, 1,000 genomes overlap, dbSNP overlap, all sorts of wonderful annotation sources. This workflow is applying that to the VCF file, and the output will be an annotated VCF file. What's cool about this is you can take this, database that VCF file, do queries on it. You can use that in your own scripts to have everything be VCF file format and have these extra annotations added as simple key value pairs is actually very, very flexible and very easy to use. This particular workflow is based on Anavar, and you can see it's actually quite complete in terms of the amount of annotation that it provides. So I'm going to click on Launch, and then we're taken to a page that describes what's running currently. And you can see this one is what we just submitted. I had one from a few hours ago that I was using in my development process. For this particular workflow, we've added some new annotation sources. And you can see I had a previous failure as I was doing the development process. What's nice about this is this will tell you what's going on. It'll tell you the status. Is it running on EC2? Is it running on the cloud? Has it failed? If you do run into a situation where you have a failure, you can email the help desk at help@nimbusinformatics.com and just provide us this SWID number. That's our tracking number that will allow us to tell you exactly what's going on with this workflow. The really cool thing is in the background what's happening is our Nimbus portal is actually launching either a single cluster node or multiple cluster nodes on EC2. And this process can take anywhere from a minute to 10 minutes, depending on the load on EC2, on Amazon's cloud, and the pricing actually of Amazon's cloud. The prices fluctuate, and we try and keep prices as low as possible, so we try and use what are called spot instances. So what's happening right now is this is literally launching a cluster on EC2 where this variant annotation process is going to run. So that's kind of the end of the process where this variant annotation is really the end of our whole genome, whole exome workflows. I'm going to show you, I'm going to step back and we're going to look back at the very beginning of the process, and I'm going to go and launch another workflow. In this case, I'm going to launch our alignment workflow. So if you look in here, there's a few different options. There's, let's see, there's the genomic alignment, or there's the FASTQ report and filter. Now we could go ahead and make a FASTQ report, and that's just going to tell us the overall quality of our samples and that sort of information. And it also lets us quality filter the samples. I'm going to go ahead and just run an alignment protocol. And in this case, this particular workflow has a lot more options than the previous workflow that I showed you. In this case, you can go in and you can fill in all this information about whether it's paired end or single-ended. We're going to choose paired end because that's what I uploaded. We can put in the read group platform unit, the sample information. All this information is really useful for downstream analysis. So I highly recommend that you fill out all this read group information that will appear in your BAM file. I recommend that you fill this out as accurately as possible. In this particular workflow, you have a choice of different standards for the scaling of the FASTQ file. I'm going to choose the standard Sanger scaling, and then I'm going to click on Next. Now what's happening here is, again, like last time, it's finding all the candidate files that I've uploaded to my account that match the particular input file format. In this case, it's gzipped FASTQ. And you can see here from last time, this is what we uploaded before. Okay, the other thing I want to point out too is this over here gives you a little bit more information about what you're selecting right now. Some workflows, including this one, have the ability to select both a single end or paired end study. In this case, in the paired end mode, it's actually going to ask me to select files twice. The first time for the first read or first read files, and the second time for the second read or read files. In this case, I'm only uploading one pair file, but you can actually upload multiple paired files. Okay, so I'm going to go ahead and select the first read now. Here's my upload event, and here's the first read, so I'm going to select that, and you'll notice it appears in my FASTQs for inputs first read file. I'm going to click on Next and wait for it to load. And I'm going to do the same process, but this time, if I click on here, it's going to tell me that this is actually for the second read. So I'm going to drill into my study experiment sample, look at the upload, and then I'm going to select the second read. Now again, you could have multiple second read and multiple first read files. The key is that they need to be in the same order. So the order of the first reads dictate the order of the second read files. Generally speaking though, most people, unless you're doing whole genome work, most people for exomes are going to have just a single lane worth of data. So you're going to have individual first read and individual second read file. At this point I can click on next, and much like last time, it gives me a summary of what is going to be passed to this workflow, including you should pay special attention to this part, which is describing the input file. So you want to make sure that your input files are correct, that this says first read, and it is indeed the first read, and this says second read, and it is indeed the second read. Okay, if you're happy with this, all you have to do is launch it. And again, you see these two workflows are now submitted. So what's happening is the cluster is launching on EC2, and these workflows will both be scheduled and run on those clusters. Okay, at this point, I'm going to end the video. And if you have any questions, please email help@nimbusinformatics.com. But at this point, we'll just let those run. And in a future video, we'll go into more details about how to string together these multiple workflows and how to pull back results.